A warm greeting to everyone. In this PPT, we'll be studying about pure culture techniques. First of all, culture is defined as a population of microbes which contains a lot of microbes, not a specific microbe. It will have a lot of population, different types of microorganisms present in it, whereas it is divided into two. Mixed culture and dual culture. Mixed culture consists of different varieties of microorganisms, whereas dual culture contains only two species of microorganisms, like that. And pure cultures or something that contains only one species, that one species which is a progeny of a single cell. And pure culture techniques are basically carried out to obtain a single species of microorganisms. The process or the steps involved in pure culture techniques basically are isolation, purification, and the cultivation of the specific species. And we'll also see about enrichment of a culture. As the name says, enrichment of a media by providing nutrients and a suitable environment for a specific microbe, not for every type of microorganism. Choosing a single species and providing the specific nutrients and a suitable environment for the cultivation of a specific microorganism. And enrichment of media were carried out by Sergei Vinogradsky, Martinez, and Van Niel in different varieties of experiments. Sergei Vinogradsky, he initially enriched certain media for specific microbial growth, and Martinez also used enrichment for specific microbes, but a specification that is without nitrogenous compounds. And Van Niel, enrichment, he used enrichment media to show photosynthetic bacteria, which actually reduced compounds as electron donors without producing oxygen. These are all about enrichment. And methods of isolation. There are several methods carried out to isolate a single pure culture, a single type of species. Those are pore plate technique, spread plate technique, streak plate technique. Initially, pore plate technique is something, the diluted sample is mixed with the melted agar, and when it is actually mixed with the melted agar, it shows a growth of multiple colonies, and which a single colony is picked, and further it is cultivated to obtain a pure culture. And spread plate technique is different from pore plate technique. Here we do not mix the sample with the melted agar. Instead, the agar that is melted is solidified, and the diluted sample is spread over the solidified agar plate. And next is streak plate technique. In this, the selected colony, specific colony that is selected, is streaked on the agar plates, and different types of streaking methods are carried out, followed to isolate a single colony of a specific microorganism. And serial dilution is also a method for isolation of a single organism. These are series of dilutions that is carried out to reduce the population of the microorganisms and to obtain a pure culture that is not yet cultivated on a solid medium, but can be cultivated in a liquid medium, and it is also carried out for the growth of a single microorganism. Next is single cell isolation. This single cell isolation is required for the isolation of selected microorganism permitted, that is, this is carried out by picking out the required microorganism and permitting it to grow. And there are two methods carried out for a single cell isolation: capillary method and micromanipulator. A capillary method is using a capillary tube to pick a single microorganism. That is, by hanging drop method, we pick a single colony from the drop of medium under a microscope and pour it into a fresh medium. That is, we culture a single selected colony and allow it to multiply in a medium. So we obtain a pure culture, single cell isolation. That is, a picked cell is allowed to grow and multiply and is cultivated. And another technique is micromanipulator, which is an instrument which is a conjunction with a microscope to pick a single cell from a hanging drop. The same as seen in a capillary method, but we use a micropipette, which is it has a micrometer which has a micropipette that moves. So using this, we pick a single drop by suction, and this drop is suspended into a medium, and then it is further cultivated for the purification of a single cell or a single microbe that is desired. Thank you.